For once, I have good news, everyone. The first human trials using cell replacement therapy for the brains of people with Parkinson's have been largely successful. In case you're not familiar, Parkinson's is a neurological disorder that affects 1 in every 100 people in the US over the age of 60. It is a progressive loss of the dopamine producing cells in your brain. You slowly lose the ability to move, and it ultimately takes lives. It can have genetic causes, although it also seems to be environmental, and until now there haven't been any real treatments, but you can replace the cells in the brain that are responsible for producing dopamine. This was shown in mice and now has been demonstrated in people using stem cells. There are 2 trials that have been effective, one of which used stem cell lines that were made prior to 2001, which is always a little bit funny because there was a moratorium on federal funding for stem cell lines. There actually still is, and the 60 that exist all have mouse cells contaminating them. This is kind of a little problem, and actually still is. When we first made stem cells as humans, we made them from embryos, and then they had to be grown on lawns of mice cells, and they would slowly contaminate the stem cells with things like RNA and DNA and mice stem cells themselves. So if you're receiving any medical treatment with older cell lines, there's a non-zero probability that you are going to get a little mouse in there. At least it worked for alleviating Parkinson's symptoms. You should know that now most stem cell lines are made from induced pluripotent stem cells, so you can take something like skin and turn it into a stem cell, and you don't actually have to clone in order to make these stem cells, which seems to be a better deal. Making new stem cell lines from the traditional method using cloning in embryos is now still, it can't get federal funding for it, although it's not explicitly not allowed, it's simply not done. Not to mention, while you can't make new stem cell lines, if you're using ones that were made recently, good luck on federal funding. So again, we now make induced pluripotent stem cells, which is a much more complicated process. This is all from a much larger area of research called cell replacement therapy, in which we take cells from healthy donors and put them into your body to help replace damaged tissues. Of course, you can also take your own cells and genetically alter them, or perhaps create induced pluripotent stem cells in order to replace components of your own self. This would have the advantage of not having rejection issues, which, by the way, might be a much bigger issue when you take older cell lines to do things like replace parts of your brain. Mind you, your brain is unlikely to reject. It could still happen. Aside the point, it is a very active area of research, and one of my favorite parts of it is where we take stem cells and grow tiny versions of organs and transplant them into people. It seems to be largely more successful than just stem cells. Yes, researchers have found that you actually need to allow cells to establish themselves before you just chuck them into a person. You're looking at a gut organoid, which, yes, has passed human trials in which you can replace damaged portions of your intestines, which is great because some of the alternatives are not so great. This can be done with hearts and lungs and kidneys and livers, but the one people seem most disturbed about is the brain. Because yes, in those Parkinson's trials, they took donor cells, not from the original person, but from a healthy donor who's able to make dopamine. But there's a question of what makes us us. I think if you're getting other cells that incorporate themselves into your brain, your original brain tissue does have to reorganize itself, so I think it's just incorporating, right? Nothing's changed. Still, at least in mice, they've been able to take brain organoids and replace quite large components of their brain with human versions of it. Yeah, that's a mouse brain with a human brain organoid. So they don't really end up smarter as far as we know when they are grown with human neurons. Right from when they're embryonic, they do end up smarter. I still think they're a mouse, so I have complicated feelings about it. How about you? I would absolutely like to be rebuilt cell by cell. My hip is still bothering me. They replaced it with some metal components. I hope you found this interesting. Follow for more, and I'll tell you about how they made brain organoids capable of experiencing physiological pain.